mild disease. And of course, we had matching controls. We isolated the plasma from these patients, and at the QBRI proteomics core facility, we have the O-link technology for proteomics. So we were able to profile those plasma samples, and collectively we profiled 920 proteins in each sample. That's a lot of proteins. And just from the first look at the data, without any statistical analysis, without any comparison, just looking at unsupervised clustering, we could easily see that the 920 proteins depicted here in the heat map actually separate severe disease from mild and controls. From here, we took two paths. The first path was to analyze proteins which are overexpressed in severe disease versus mild and controls. And I know this map is complicated, just briefly to explain. In these red dots, we have the proteins we discovered from the profiling that are quite high in the plasma from patients with severe COVID complications. And we have in gray protein-to-protein interactions, which actually explains how complex the response of the host, our body, to the coronavirus. In blue, we have drug-protein interactions. So we looked for FDA-approved drugs that interact or inhibit those proteins that are highly upregulated in the plasma of severe COVID. There's a lot of information here, and I thought the best way to summarize it is to look at drug classes. So obviously we have the antiviral drugs, but we also have anticoagulation drugs. We have the glucocorticoids, which was shown to be effective in the recovery trial. We also have some chemotherapeutic kinase inhibitors, and of course we have immunosuppressive drugs. So there was more than 300 drug predicted by this analysis. So we still have to know, but it's comforting to know that the dexamethasone, which was shown to work and reduce mortality, came up in our study. Aspirin also came up, and this is actually entering the recovery trial as of 6th of November. The announcement was made. And we have other drugs that shown success in clinical trials, phase 1 or phase 2. So there's a lot of work to go and understand this and actually come up with something that's effective. But my opinion, and it's not really my area of expertise, I would think maybe one drug from each of these circles might work together in combination. Who knows? The next path we've taken is to actually see if we can develop a predictor, a molecular predictor that does precision prediction of a patient's risk of developing complications. And we've analyzed the 920 proteins, and we found 46 protein signature. It's shown here. It's composed of two modules. We calculate a score for each patient based on this expression. Each red colored dot is overexpressed and the blue ones are underexpressed. In summary, we found that the score calculated signature actually differentiates severe COVID cases from mild cases and controls, and also mild cases versus controls. We wanted to be more comfortable at this, and luckily, the MGH with the Harvard and the Broad Institute have actually done a study, and they made it public. And they did use the O-link study on a group of patients. So the nice thing about the study, they have actually looked at longitudinal. So basically, when patients first went to the emergency departments because of respiratory distress or other similar symptoms causing COVID suspicion, they got tested for coronavirus. Some of them were positive, and they continued enrolled these patients in this study, taking blood on day 3, day 7, and day 28. We calculated the signature we have. We call the molecular severity score, exactly how we developed it from the Qatari population or the Qatari study. And we found that in patients who die eventually within 28 days because of COVID, or patients who get intubated to receive oxygen but survive for 28 days. So these would be defined as severe cases according to WHO cardinal outcomes grading. So we can see our severity score actually is much higher than controls or patients who did better, and increase over time, particularly for the patients who died, but then reduces over time, particularly after day 7. So we tested the performance of our test in this completely independent cohort, and we find that scores that are calculated on day 3, so if you collect blood on day 3 or day 7, they're quite predictive of severity. So from here, what we're planning to do, our next step is to do a clinical validation trial to test the robustness of this prognostic signature, most likely applied on day 3 to allow decision-making for clinical care. Who are the patients are most likely to develop complications, intervene earlier, be ready for them, and who are the patients who would not develop severe complications. So at the end of my talk, I would like to acknowledge my collaborators, particularly in this paper that we're publishing, particularly Dr. Mariam Al-Nas from HMC, and of course we have a lot of collaborators the color coded according to different organizations that included HMC, the CDC, the ADLQ, and of course Qatar University and QBRI. Thank you all.
Thank you so much, Dr. Faris, for that very succinct, very clear presentation. Can I ask, for your controls, can you distinguish between patients with different infections, like bacterial pneumonia in sepsis compared to COVID-19?
Yeah, that's a very good question. Yeah. So we were lucky again with the MGH cohort. That cohort actually had people who came to the hospital with respiratory distress. About 74 of the people who presented with those symptoms did not have COVID, but they still included them. And the answer is yes. We can differentiate at time 0. Unfortunately, they only did day 0. We can differentiate between COVID and non-COVID. And that's the diagnostic side of my presentation, but I didn't want to take too much time explaining it. But the answer is yes. There's an increase of the signature with the patients who are not COVID, not SARS-CoV-2 positive, but it's not to the extent as to SARS. So I would say the signature might be prognostic or diagnostic for the disease itself, but more so when it's because of SARS-CoV-2.
And do we have an indication that some of these proteins would be risk predictors for, say, progression to really severe symptoms compared to maybe even regression and return to good health?
Some of them, yes. I mean, I'll tell you one of the proteins is ACE2, not surprisingly enough. That's one of them, for example. And IL-6 is another protein. The answer is yes. And the trend over time is quite predictive. So if I would have the luxury of running this test completely in a diagnostic lab, I would say do a test on day 3 and day 28, and maybe in between. And you can actually find out the way it behaves over time is predictive, not just one particular time points, but also, and that's a very important question, and we don't want to include in the current manuscript we're finishing, is to actually look at agonists of the downregulated proteins, because some of them come to my eye as something that may be protective, so it could be preventative in that sense.
Okay. Thanks so much. So now time is moving on, so maybe we can move on to the next presentation by Dr. Noor Majbour, who's a research associate in QBRI and on serology test for COVID-19. Dr. Noor, please.
Thank you so much. Thank you. So in my presentation, I will describe the work that we have done developing an in-house laboratory test that can help us estimate how many people in Qatar have been infected with SARS-CoV-2 virus, the virus that causes COVID-19. So the test is called the serology test. It's also known as an antibody test. So as you can see on the surface of the virus, there are several proteins that are expressed. We have mainly focused on the S1 and RPD domains of the spike protein, as well as the nucleocapsid protein. And as highlighted by Dr. Faris, we have also looked at the cohort from HMC, including control subjects, as well as patients at mild and severe stage. So our serology test is ELISA-based. So following sample collection and serum separation, all the proteins were coated on the surface of the ELISA plates, and then the serum samples from the patients were incubated with the viral proteins. So if the patients have antibodies against those viral proteins, they would form a complex, and that will generate a color-based readout. So in our serology test, we have mainly focused on the levels of the IgG and IgM antibodies in those serum samples. So the heat map shows the count of the serum IgG against all 3 proteins, the S1, RPD, and N individually and combined. And the color and the heat map intensity, the intensity of the color reflects the levels of the serum IgG. So the darker the color, the higher the levels. And as we can clearly see, either through the heat map or the bar graph, that the levels of serum IgG were much higher in patients, both mild and severe compared to control. And we can also clearly see that the levels were even significantly higher in severe patients compared to mild patients. And Similar observation was also noted in the serum IgM levels in the same cohort. So we can also see that the serum IgM levels, were significantly higher in the patients compared to the healthy controls. However, as you might have noticed, that the overall levels of IgM antibodies are lower compared to IgG, but this is also expected given that IgM peaks at the very early stage of the infection. So to estimate the discriminative power, the sensitivity and the specificity of serum IgG levels discriminating between the patients and the healthy controls, ROC analysis was carried out, and as you can see, the rates of sensitivity and specificity were above 80%, and with AUC that is above 0.9, which is considered, very strong discrimination when control subjects are compared to severe patients. Similar but lower levels of sensitivity and specificity were achieved comparing mild patients with control subjects. Now, to further validate our findings using our in-house serology test, we have explored the correlation between the data that was generated using our ELISA serology test and the data generated with Syngenix technology, more specifically using the ImmuSafe from Syngenix, which is a lab-based biochip that also measures the levels of the antibodies against SARS-CoV-2 protein. And here we look at the correlation between the levels of the IgG, serum IgG levels against the N protein, and we can clearly see that there is a strong positive correlation between the data generated with our in-house ELISA and the one from Syngenix in both mild and severe patients. A few words about our recent assay, the neutralizing assay. It's slightly different from the serology test, meaning that the neutralization assay will not only tell the researcher if the patients have active antibodies, but also will tell the researchers if the patients have effective antibodies against the virus. So the patient would have to have antibodies that can actually bind to the cell entry protein and thus block the binding of the virus to the cell and thus block the infectivity of the virus. So what we have done, we have actually expressed in-house the RBD protein. We have also expressed the ACE2 protein that is exposed on the surface of the cell. And we have also generated a nanobody. And what we have first showed in our neutralizing assay, the specificity of the RBD protein binding to the ACE2 protein. And then next, we validated the specificity of our in-house nanobody binding to the RBD protein specifically. And we also have showed the preliminary data showing that the antibody could actually bind to the RBD protein, blocking its binding to the ACE2 protein, showing that there is, so I can, say that our neutralization assay is now ready to be screened against the patients from samples to look for the ones who actually have these neutralizing antibodies. With that, I would like to thank all the patients, the volunteers, and our partners, and as well as the funding agencies. Thank you.
Okay, thank you so much, Dr. Noor, for that very interesting presentation, very clear, interesting presentation. You see increased levels of the antibodies with increasing severity, and you showed a correlation, I think, with the nuclear protein. So does this mean then that effectively there's a correlation between the antibodies and viral load, do you think, in patients?
Well, I think I can't really say if the correlation with the viral load, but it's certainly with the immune response. So, I cannot really, decide at this stage, because the severity, we are referring to the severity of the symptoms. So I don't know at this stage if the severity of the symptoms was actually correlated with the load of the virus, or maybe because those patients had, other comorbidities, and that's why they had, more aggressive immune response.
And is there any association between the level of antibodies and the prognosis of the patient, clinical outcome?
So this is absolutely a very good point, and this is something that we are planning to look at. So I mean, this is what we have done so far was only, I mean, the test was only at a single time point, and I have not yet analyzed the data. I mean, in respect to the clinical profiling of the patients.
Can you briefly? So maybe Dr. Prasanna-Kalotka, please proceed with your presentation.
Okay. And so my presentation is showing a nice collaboration between the Qatar Computing Research Institute in Doha, as well as QBRI, which I'm a part of. And so it's been a really good team with QCRI and QBRI, and thus the information I'll be talking about in the next 5 minutes. And it's about doing repurposed drugs that were essentially predicted by the AI algorithms developed by QCRI, and we will be doing the validation. And so, of course, to start off with, a quick idea of what COVID-19 is, obviously, is that it's something like about 29 proteins, that's all that make up this entire virus. But in there, in our study, we're going to be focusing on 3 of the most critical bits. Okay, we are going to be focusing on 3 of the most important proteins. And so we're looking at, we're going to be looking at obviously the PL-PRO, which is a protease associated with both replication as well as the innate immunity response and its inhibition. Also, the 3CL-PRO. This is an enzyme which is the key cleavage enzyme that breaks up all the polypeptide chains into the individual protein subunits. And finally, and not least importantly, of course, is the spike protein and really the RBD, or the receptor binding domain. And those were the 3 specific areas targeted by the people at QCRI. And their team at QCRI developed this flow. So what do? They take several different databases that have information on the sequences, but in those databases, there's also correlations with binding affinity of different drugs and compounds that are available in these other chemical databases. And so they essentially set up a framework for not just COVID, but all viruses. But in this specific study, then they narrowed it down and looked at only how these specific proteins that I just spoke about in the previous slide are going to be targeted, as they have the most information, including things like high resolution 3D structures, which can then be validated by other techniques, including what I'm going to be talking about of what we did with QCRI. So once they had come up with these 3 targets and these repurposed drugs, and as you notice, the hydroxychloroquine wasn't one of their top hits, but these standard antivirals were. But what was more interesting is that when we also did the docking, then this is in silico docking, computational docking, what we found out that something called rifabutin, it is not an antiviral, it is an antibiotic, made up some of the best hits in terms of what we see as binding to all the 2 proteases as well as the spike protein. And in fact, their AI predictions from QCRI also said that rifabutin should be a particularly good binder, and this was somewhat unexpected. However, obviously, we will need to do some more validation of that, and I'll be talking about that on a later slide. What's interesting, though, is besides rifabutin, which you can see tightly binding in all these proteins, I can't go into the details in 5 minutes, but remdesivir, a very well-known compound, obviously, which is an antiviral, it didn't bind any of these proteases nor the spike protein. Now that's more or less as expected. The remdesivir is thought to bind the RNA-dependent RNA polymerase, so you wouldn't really expect it to bind these. So to us, it also shows that this was served as a very good control, that these algorithms and AI aren't just randomly predicting that any sort of antiviral would bind any sort of our protein. So we believe there's quite a bit of specificity to our predictions. And what I've talked about so far has been completely and absolutely in silico. So it's all nice and good what you predict in silico, but in our lab, the future direction, the very near future direction, will be actual wet lab validation, and we do that with something called isothermal calorimetry, or ITC. It's nothing more than just a really fancy and expensive thermometer. What it measures is anytime you take two things, let's say a protein and a drug, and you put them in the same cell, there will be a reaction if there's good binding, and that will give off an energy or a change in temperature. And based on what that shape of that temperature change and how fast it is, you can tell not only how avidly or how tight the binding is, but also what are the determinants of that binding? What are the potential side chains? Is it hydrophobic? Is it hydrogen bonds? What is creating that? So it gives you a real good profile of that binding. And finally, I just want to thank the main people involved with what we're doing. And so far, it's been Zial Islam, a very good postdoc in my laboratory, and we had a very good partnership with the QCRI team. And this work, all the way to the docking, is currently in bioinformatics. It's under revision. And in the very near future, I mentioned we'd be doing isothermal calorimetry. Now here I am thanking Dr. Swain Chen, who has already given us some plasmids to start making some of these COVID proteins in the near future. But what I should have also said that, as Dr. Noor had just mentioned a few minutes ago, Dr. Omar's lab is actually also making quite a bit of these COVID proteins. So there's a lot of synergy within the QBRI, but more importantly, we also see synergy with QCRI. Whether these compounds will be useful for COVID or not is interesting, because there's a lot of vaccines coming out. But what's nice is we've set up a very good framework for the future where the computational and the wet labs are working together. And I thank you.
Thank you so much for that presentation, Dr. Prasanna. Sorry I missed some of that. I had to drop out because of the poor connection. So I noticed at the WISH 2020 symposium there is a session by a consortium that is involved in doing molecular graphics-based drug development against COVID-19. So how are other people have addressed this issue in the search for new therapeutics for COVID-19?
Well, I don't know about COVID-19 specifically, but obviously the whole idea of in silico docking, AI predictions, they've been pretty much at the forefront of many different labs. I mean, I think everybody really remembers the big hit stories like in the HIV and other specific viruses where they found really good binding or inhibitors. What's important about things like the graphics-based approach is you not only get a good idea of what the binding is, because you know what the atomic or molecular determinants are, you can then improve that binding by making better molecules, which is what was done with some of these other viruses. But whether we do that for COVID or 19 or not, I guess that'll be remaining to be seen.
Dr. Faris, Dr. Noor, would you have any questions for Dr. Prasanna? Maybe.
Study, I'm actually quite keen to look at cross-checking what you're finding as predictions and what I found, because I think the antibiotic actually saw it in my list. I'm just checking now when you're talking about it. What's the name of it? The antibiotic?
Rifabutin. It's against tuberculosis and mycobacteria.
Yeah, actually. I got a lot of antibacterial, not just antiviral, and I did not understand why. Maybe you can explain why. So we should talk about this soon, I think. Right.
Okay, excellent presentation. So maybe we can pass on to the final presentation now. My presentation. Is the presentation clear to everyone?
Yes.
Okay, so I'd like to thank the organizers of Virtual Wish 2020 for inviting me to present at this symposium. And I'd like to talk about anti-cancer drug repurposing for treatment and dietary supplement for improved respiratory health and prevention of COVID-19. So I've been particularly interested in looking at the vulnerabilities of the SARS-CoV-2 virus to proteotoxicity, and this may provide an opportunity for repurposing of existing drugs for treatment of COVID-19. Drug-induced proteotoxicity is a strategy in antiviral drug development where the concentration of endogenous reactive metabolites are increased to virucidal levels. For looking at reactive metabolites, there are mainly two options. There's reactive oxygen species, ROS, which target cysteine, methionine, tyrosine and tryptophan residues in proteins and cause oxidative damage. And then there's the reactive glycating species, such as the most reactive is glucose-derived methylglyoxal, which targets arginine residues within proteins for modification. Which one to try to increase depends on which amino acid residues are enriched in functional domains of the SARS-CoV-2 proteome. And we have a way of analyzing this. We have a bioinformatics tool called the receptor binding domain analysis, whereby we can identify amino acids in functional domains of the SARS-CoV-2 proteome. And when we looked at this, we found cysteine, methionine, tyrosine and tryptophan were actually negatively enriched or had an enrichment factor less than 1. So actually they're depleted in functional domains. SARS-CoV-2 is resistant to oxidative damage, but arginine was enriched 4.9-fold actually, and also enriched higher than the proteins of the human host. So this means the SARS-CoV-2 is sensitive to damage and inactivation by methylglyoxal. And before the COVID-19 pandemic developed, we'd already identified drugs that increase methylglyoxal in cells to virucidal levels. And some of these are anti-tumor drugs. So this provides a strategy now for looking at anti-tumor drugs and their anti-SARS-CoV-2 activity. In the next slide, I show in a scheme how we envisage this playing out. There are two key proteins in the SARS-CoV-2 virus that are sensitive to MG modification, the spike protein and the nuclear protein. Modification of the spike protein by methylglyoxal would block infectivity by the virus, and if any MG modified virus was produced during drug treatment and leaks out of cells, it's then an inactivated virus and would behave like a vaccine, an inactivated virus vaccine to increase host immunity. Modification of the nuclear protein by methylglyoxal prevents viral assembly and then terminates virus replication. So these are really two important responses and could mediate effective treatment by drugs that can increase methylglyoxal. And the two drugs that we identified were shown there, doxorubicin and paclitaxel. And we're now evaluating these drugs against live virus with collaborators in Qatar University. Secondly, that we have the problem is how can we prevent COVID-19 and be better prepared to resist future waves of coronavirus respiratory infection? A key risk factor for susceptibility to SARS-CoV-2 infection is age-dependent decline in lung function. And here I show in the slide the trajectories of lung function decline in men and women. And you can see above the age of 60 and 80, that's when our lung function starts to decline. And this leaves us susceptible to respiratory infections. Actually, the drivers of age function decline are hyperactivation of cell-mediated immunity, increased production of monocyte chemoattractant protein 1, MCP-1, and interleukin-8, neutrophil chemoattractic protein 1, that leads to increased recruitment of monocyte-derived macrophages and neutrophils into the lung of elderly people. They're activated and then they trigger fibrosis and stimulate decline in lung function. So the problem is, how can we prevent decline in lung function? How can we prevent the decline in lung function in the elderly? And the solution, I suggest, is an evidence-based dietary supplement to decrease MCP-1 and interleukin-8. Dietary supplement, because dietary supplements are very safe and are appropriate for long-term administration, such as this application for prevention in an elderly patient group. And before the pandemic developed, we optimized a dietary supplement and evaluated it in the Healthy Aging Through Functional Food study, which was optimized to reverse insulin resistance for prevention of type 2 diabetes. In a randomized double-blind placebo-controlled crossover clinical trial, we gave the supplement oral capsule once daily of 90 milligrams trans-resveratrol and 122 milligrams hesperidin. These are compounds found in red grapes and citrus fruits respectively, but are given here at doses higher than you can achieve in the diet. And the remarkable finding was that this supplement decreased interleukin-8 and MCP-1, the target inflammatory mediators we're seeking if we want to try to prevent lung function decline in the elderly, by 31% and 49% respectively. This is measured in peripheral blood mononuclear cells, PBMCs, but we would need to see if we get a similar response in lung alveolar epithelial cells. But now it's a target, it's a potential dietary supplement to repurpose to the elderly to try to prevent decline in lung function and provide resistance to developing COVID-19 and other respiratory infections. So key messages are SARS-CoV-2 coronavirus appears vulnerable to modification by reactive metabolite methylglyoxal. Anti-cancer drugs increasing Mg are being evaluated for therapy of COVID-19. Dietary supplement trans-resveratrol hesperidin may be repurposed to prevent lung function decline and provide resistance to COVID-19 in the elderly. And as vaccines for COVID-19 emerge, we need to maintain our impetus in antiviral drug development and respiratory health prophylaxis for future resistance to respiratory infections. Then I'd like to thank my research team working with me in the MG and glyoxylase research group shown there, Dr. Mingzhang Zhu, Dr. Alberto Lidafuente, Dr. Patrick White and PhD students Mariam Al-Motor and Aldan Al-Nefs, and also our collaborators at Qatar University, Dr. Naila Rabbani, Dr. Heba Al-Khatib and Dr. Hadi Yassin. And I'd like to thank Qatar Foundation and Qatar University for funding, and also you for your attention. Thank you so much. So are there any questions? There's no questions.
Dr. Paul, you mentioned that MG and MGO, you could use anti-cancer drugs to potentially increase them. Are there any natural products that also have large amounts of these, or would it have to be a drug?
Yeah, that's an interesting question. I mean, we need anti-cancer drug-induced increase in methylglyoxal because they increase the flux of methylglyoxal production. People have, it would, for example, if you were to give a bolus of methylglyoxal orally, it would be metabolized so effectively by human tissues, you wouldn't be able to increase it. But these anti-cancer drugs have the ability to increase flux within inside cells and raise the cellular level. We also have an experimental drug, which is called a glyoxylase 1 inhibitor, which inhibits the main enzyme of methylglyoxal production, and it can have potentially antiviral activity, and has been studied for anti-cancer activity in the past. So we have these studies ongoing with the virology researchers in Qatar University. It's too early to say what the outcomes are, but it's looking very promising. And maybe we will stimulate other people to explore this interesting area of producing proteotoxicity to the SARS-CoV-2 virus. And of course, we're all hoping that MG modification turns out to be the Achilles heel, the weak point, or one of hopefully more than one weak point of SARS-CoV-2, so we can get effective treatments for COVID-19. It's also interesting about this decline in lung function in the elderly, I think, which probably underlies a lot of predisposition of the elderly to COVID-19. And the elderly, we know, over the age of 65, 70 are the people at most of risk of severe disease. And about 54% of the variation in lung function decline is controlled by genetics. So there's quite a strong environmental factor. So it would be difficult just by genomic sequencing alone to identify people at risk, I think, of relatively rapid lung function decline. There seems to be other environmental factors that maybe could be picked up in a proteomic abundance screen, like as Dr. Farris talked about in his presentation. But I think that is also very interesting to try to to develop prophylaxis so we get increased resilience of our vulnerable populations in the future to future waves of SARS-CoV-2, which I hope there won't be many, or other coronaviruses that may jump species barrier in the future. But we need to be prepared, and with treatments, with better diagnostics and prophylactics, inshallah, we will be better prepared next time. So are there any comments? We have a few more minutes to go before the end of the session.
Yes, Dr. Paul, I think we have a couple of questions, actually. The first question, I think, for PK. What can be the possibility of rifabutin in COVID drug use?
I mean, I think that's a good question, and that's what we were wondering when we came up with these results. And I think Dr. Fares is probably wondering the same thing, given his results. And my feeling is that obviously further validation is needed. I don't think we need to do another hydroxychloroquine. I mean, I think we need to be careful, because as we start developing a set of drugs that has some efficacy, I think any new entrant should already be quite validated. Having said that, it's an FDA-approved drug, so it's most likely already going to have a lot of the toxicity and other screens that it's passed. So perhaps it could be introduced relatively fast. But again, I would do other validation, including binding. What I didn't have a chance to say earlier was cellular assays, and I'll leave it at that.
And that actually goes the same, because so the second question, it asks about repurposing. I'll come back just to follow on with PK. Drug repurposing, it's usually you follow FDA-approved drugs, so you don't have to do a clinical trial, particularly when you're trying to do quickly. But also there's prior information. For example, I mentioned I saw in the network analysis I've done 300 drugs. I mean, that's a lot of drugs. Which one is right? And the first thing I noticed, just reading about the drugs, some have contradictions with others. So clearly, pharmaceutical and medical information has to come in before doing anything like this. But also what both Dr. PK and Dr. Paul mentioned, we are in collaboration to do these, infection assays in vitro to actually understand it, to really characterize it and then go to trials. And this all sits in a longer term resilience plan. I mean, God forbid, but let's say we got SARS-CoV-3, right? It's possible. We need to get better at treating the disease, because we might need a new vaccine or something goes wrong. Again, God forbid, but we need to start learning how to treat the disease itself. So the viral pneumonia and the ARDS, the acute and severe respiratory syndrome. So that's what we need to get better at. And clearly, with the repurposing or the work done with the recovery trial, it had an impact globally. So I don't know if it was in Science before the weekend. In the US, mortality has decreased by 30% because of better treatments. So treatments are important. The last thing the second question also included, there are multiple factors relating to COVID severity and certification, and larger validation is needed. Could not agree more. The study we did, we actually corrected for BMI, for obesity, for many things. And we've also discovered some clinical parameters that were available because HMC have done a great job by collecting all the clinical information. And we find things that no one has considered them before, and things that people have just realized, like neutrophil counts, it's correlated with severity. And the point from all of this work, I mean, we've done a study with 100 cases, the MGH, the full data, had 306 cases, and this cross-validation is comforting. But yes, of course, we're planning to do another follow-up study to actually, the first ones were discovery, now we're looking at validation. And the idea is to try as soon as 3 days, 4 days, maybe, or symptoms, do this triaging of patients, high risk, low risk.
Yeah, there are some questions coming in now, actually. One is, how does the antioxidant like vitamin C or zinc help in prophylaxis? Well, that is a very interesting, because in the very early days in Wuhan in China, there was very high dose vitamin C given, very extremely high dose, infusions, I think equivalent to giving about 20 grams a day. And in initial open trials, there was some suggestion maybe of benefit, and they were doing then a randomized a blinded trial, I think, to finish in September this year. I haven't heard what the outcome is. The issue is really over efficacy, and usually if you get more than, say, I think 200 milligrams vitamin C dose a day, it's washed out in urine, so you don't get any health benefit above 200 milligrams. And it's maybe not conceivable that we could go. Well, we need to see if this very high level vitamin C infusion has any effectiveness in randomized trial. And probably if it does, it's working by something other than an antioxidant level mechanism because of this very high loading effect. And then question 4, do you think aerobic fitness of lungs through exercise have any bearing on health in the elderly? I guess that's a question for me. I think, of course, as we're all encouraged to have a good diet and do regular exercise to keep healthy in later years, then I think it could help. I mean, of course, there are some obvious things that lead to deterioration of lung function. One, of course, is smoking and other asthmatic-like exposure to pollution and develop a sort of asthmatic-like hypersensitivity responses in the lungs, and prior serious infection with, say, and development of pneumonia. They all are risk factors for declining lung function. But so if we could get a really good prophylactic and help our elderly maintain stronger lung function, it could probably provide some resistance to help them be more resilient to respiratory infections. So we're coming to the end of the session now. They say the session will be extended 5 minutes. So would we all like to make some concluding remarks briefly? Dr. Faris? Yeah,
so I mean, COVID has been a very hard experience for everyone, but it actually was good collaboratively across Qatar Foundation entities. We all have good collaborations with many different places and built relationships. And these are important for the future because the coronavirus have become like the flu now. I don't want to scare anyone, but we have to be careful, resilient, and prepared. So all the things that we have been doing is targeted towards this. And even if a vaccine comes, it might take some months before it's distributed and there's enough doses for everyone on the planet. Mm-hmm. And we also have to be ready for the future. We don't know, and it's been a waking up experience. And I would love to see that what we discovered, at least from the projects I've talked about with HMC, is actually get it validated and move forward, because early prognostication might do a lot in terms of earlier treatments.
Dr. Noor, your concluding remarks, please.
Well, I think maybe the thing I enjoyed the most, was how we were able to redirect our expertise and our know-how to a different field. I personally have no background in virology at all. My background is mainly antibodies and assays development and protein chemistry. So it was, a very good learning experience for me to apply that expertise for a different purpose and a different cause.
Okay, thanks. Dr. Prasanna, your concluding remarks.
Just 2 quick ones. One is it really has shown us how much in shape we should all stay. It's not about aging. It's not about anything. And we don't need doctors. We need ourselves to really have a healthy lifestyle. And the second thing is, as everybody has said, Dr. Noor, Dr. Faris, and yourself, I think we're really doing great collaborative work throughout Qatar. If not COVID, we'll be ready for the next onslaught, and it will come.
Yeah, I've been really impressed by all colleagues in QBRI, how they've turned their ingenuity, experience and expertise into tackling this unprecedented issue of the COVID-19 pandemic. And we've all had expertise that can help fashion, I think, improvements in diagnostics, repurposing of therapeutics, and maybe look towards also prophylactics to provide populations with improved resistance and resilience to this type of infection. Inshallah, we'll get good progress with this, and hopefully we can push back very strongly against COVID-19 and any similar coronavirus infections that make a species jump to pose a threat again. So I think we're almost at the end of the session now. So I'd like to thank you all so much for your presentations. They were so beautiful slide presentations and nice commentaries on your research. And thank the people who sent us in questions, very interesting questions. And if there's any more things you would like to follow up, please contact us at QBRI. You can find our contact details on the QBRI web pages. We'd be very keen and interested to hear from you. Thank you so much.
Thank you.
Thank you. Bye. Assalamu alaikum.
Bye.